Welcome to this explainer. Today, we're putting on our detective hats and diving straight into the really fascinating, kind of invisible world of food forensics. We're looking at a groundbreaking 2026 study that honestly reads less like a dry scientific paper and way more like a high-stakes true crime story. It's all about uncovering the invisible truths hidden in our everyday lives and the scientists who acted as lead detectives to solve a literal billion-dollar mystery. We're going to look closely at the story behind this research, the brilliant, obsessive detective work that actually made it possible to track down exactly what we're putting into our bodies. I am absolutely thrilled to guide you through this.
So do you know what's really in your marshmallows? It's a super simple question, right? But think about it for a second. Globally, we humans consume hundreds of thousands of tons of gelatin every single year. When you grab a bag of marshmallows or those chewy gummies you love, it's the gelatin giving it that perfect, bouncy, melt-in-your-mouth texture. But hidden inside that ingredients list is a surprisingly complex mystery. On the label, it just says gelatin. You literally cannot tell if that gelatin came from a pig, a cow, or a fish just by tasting it, right? A strawberry gummy just tastes like a strawberry gummy. But to a food forensic scientist, that single word, gelatin, is a massive chemical puzzle just waiting to be solved.
Here's our roadmap for cracking this case today. 1. The hidden gelatin problem. 2. Introducing Safani 2026. 3. The amino acid fingerprint. 4. Statistical sorting hats. 5. The forensic verdict. And finally, 6. The future of halal. Let's get right into it. All right, Section 1. The hidden gelatin problem. Unmasking a massive industry. To really understand the science here, we first have to understand the business. And the global gelatin manufacturing industry is massive. We are talking about a market worth a staggering $1.34 billion. And here's the crazy part. About 80% of the products produced in this massive industry rely on hydrolyzed skin gelatin. Why? Well, because it's an incredibly cheap and abundant byproduct of the global meat industry. But, with that sheer amount of money on the table, the financial incentive for food fraud is astronomical. There's just this massive temptation for shady suppliers to cut corners, like taking super cheap pig gelatin and secretly passing it off as much more expensive premium bovine or cow gelatin.
And because the financial stakes are so unbelievably high, this kind of food fraud becomes a very real temptation, which of course creates huge, genuinely terrifying concerns for consumers worldwide. We're not just talking about a slight dietary preference here, it's about absolute necessity. You've got massive public health concerns, like the very real terror of transmitting bovine spongiform encephalopathy, mad cow disease, from tainted beef products in the supply chain. Then you have millions of consumers who strictly require halal or kosher foods for their religious beliefs. It is a profound breach of trust for someone keeping strict kosher or halal to unknowingly eat mislabeled pork products. And that's not even mentioning vegetarians who want to avoid animal products entirely. They all absolutely need to know, without a shadow of a doubt, does this gelatin come from a pig, a cow, or a fish?
This brings us to Section 2, Introducing Sani 2026. Enter the investigators.
Facing this deeply flawed global supply chain where labels frankly just couldn't always be trusted, a dedicated team of researchers decided they had to step up. Existing testing methods were largely failing the broader public because they were way too complex and mostly limited to wealthy nations. So, lead researcher Mohammed Shirwan Abdullah Sani and his investigative team set out to develop a brilliant, foolproof way to catch food fraud in the act. In the 2026 book, Beyond Halal, they published their groundbreaking findings in Chapter 16. This basically serves as their manifesto for a better, more transparent system. Their goal was incredibly ambitious. They wanted to unveil a completely new, widely accessible, and hyper-reliable approach to authenticate the exact animal source of skin gelatin literally anywhere in the world.
Now, the old way of doing this involved machines called mass spectrometers. These machines are highly sensitive for sure, but they're essentially the supercars of the laboratory world. They're incredibly expensive to buy, they need specialized fuel, and they are even more costly to maintain. They act like these massive, ultra-precise weighing machines. Sani's team wanted to completely democratize food testing, so they actually ditched those expensive mass spectrometers in favor of a much cheaper, highly effective alternative called a diode array detector. Think of this new tool not as a massive weighing machine, but more like a specialized camera that uses different wavelengths of light to identify chemicals. They theorized that what they lacked in multi-million-dollar hardware, they could totally make up for with clever chemistry and advanced statistics.
Moving on to Section 3, the amino acid fingerprint, finding the chemical clues.
Okay, so to use this specialized light camera, the researchers needed a clue. They needed a chemical fingerprint entirely unique to each type of animal. And nature, as it turns out, leaves its own hidden barcodes. By breaking the gelatin down using acid, the researchers separated it into 17 distinct amino acids. You can kind of think of this process like taking a massive, complicated Lego castle and pulling it completely apart to count the exact number of red, blue, and yellow bricks to figure out exactly who built it. They discovered that specific concentrations of these amino acid bricks actually act as a unique chemical signature. Just look at the staggering 33.66% concentration of glycine in porcine or pig gelatin right alongside those specific levels of proline and hydroxyproline. A cow's gelatin will have a totally different ratio of bricks, and a fish's gelatin will be different again. This specific ratio, that was their smoking gun.
Which leads us to Section 4, Statistical Sorting Hats, making sense of the clues.
Because finding the chemical clues is one thing, but human brains simply cannot visualize 17 different dimensions, like those 17 amino acids all at once. How do you sift through thousands of complex chemical data points to confidently identify the source without just getting lost in all the noise? Well, you need a powerful way to organize the evidence. This is exactly where Sana's team brought in the heavy mathematical artillery, multivariate data analysis. Standard math just wouldn't cut it here. You can really think of MDA as a highly advanced digital sorting hat. It takes all those complex, overlapping, multidimensional chemical fingerprints and mathematically groups them based on hidden similarities that the human eye could literally never see on its own.
To make this data actually visible to us, the team used a specific type of MDA called principal component analysis with Varimax rotation. Now, I know Varimax rotation sounds incredibly dense, but it's actually pretty intuitive. Imagine looking through a kaleidoscope where all the colorful pieces are just totally jumbled up. Varimax rotation is simply the mathematical equivalent of twisting that kaleidoscope until all the colors suddenly lock into perfectly clear, understandable shapes. It was an absolute aha moment for Sana's team. The mathematical model visually separated the pig, cow, and fish gelatins into beautifully distinct, undeniable clusters. The pink, green, and blue groupings emerged to show that the chemical fingerprints were distinct enough to be isolated with zero overlap. It's just brilliant.
All right, Section 5, the forensic verdict, cracking the case.
So the detectives had their chemical clues and they'd perfectly tuned their digital sorting hat. But the tension in literally any investigation comes down to the trial. Would their predictive algorithms actually work in practice to catch food fraud? They tested several different models to see which one could best predict the animal source purely based on that raw amino acid data. And guys, the results were a massive victory for food science. Their chosen model, called discriminant analysis, achieved a spectacular 100% correct classification rate when sorting the training data into porcine, bovine, and fish categories. It didn't make a single mistake. 100%. It was completely flawless.
To be absolutely sure they had the right tool for the job, the team actually compared their champion, discriminant analysis, against another popular model called partial least squares. Now, while the partial least squares model was quite good, it hit a 93.3% accuracy rate on the testing datasets. Discriminant analysis proved undeniably superior. Not only was it more accurate, but it also had significantly lower variance. In plain terms, variance just means how loosely packed the data is. Discriminant analysis didn't just sort the animals correctly, it packed them so tightly together in the data model that there was absolutely no gray area, no wiggle room, and no confusion. It cemented discriminant analysis as the definitive, foolproof forensic tool for this entire investigation.
Finally, Section 6, the future of halal, a new standard for food transparency.
So, what does all this incredible detective work actually mean for the future of our food supply? Well, it is nothing short of a paradigm shift. As the researchers state in their conclusion, this method really paves the way for widespread global adoption. Because Sanya's team intentionally designed this method to use that much cheaper diode array detector, a food testing factory in a developing nation can now actually afford to run these tests. This completely democratizes food safety on a global level. Authorities worldwide could realistically adopt this as a standard everyday test to authenticate gelatin products. It ensures that when a label promises something is bovine or halal, it is scientifically proven, beyond a shadow of a doubt, to be exactly that.
Sanya and his team successfully journeyed into the murky, multi-billion-dollar underbelly of the gelatin industry and turned it into a cleanly solvable math problem. They've shown us that the invisible world of food chemistry can be made visible, affordable, and incredibly precise for everyone, everywhere.
And it leaves us with a fascinating, highly personal thought. As these powerful forensic tools become mainstream, stripping away the mystery of mass-produced ingredients and forcing total accountability onto manufacturers, how is this newfound transparency going to change the way you look at your next bag of marshmallows? Thank you so much for joining me on this deep dive into the incredible world of food forensics.